Ever stop and think about how you work? I mean, how do the trillions of cells in your body know exactly what to do, second by second? It all comes down to a secret language, a microscopic instruction manual that life uses to write its own story. Well, today, we're going to break that code.
It sounds kind of crazy, right? The entire blueprint for you, me, and every living thing is written in a language that has just 4 letters: A, T, C, and G. So how on earth does that incredibly simple alphabet create the staggering complexity of a human being? That's the puzzle we're here to solve together.
So, here's our game plan. First, we'll get down the fundamental rulebook. Then, we'll follow the instructions through 3 absolutely crucial stages: making a copy, writing a message, and finally turning that message into a finished product. And by the end of this, you'll be able to crack the code all on your own.
All right, let's dive right in. Our journey starts with the master plan itself. In biology, there's this core principle that explains how all genetic information flows, and it's called the central dogma.
Think of it like this. The central dogma is basically a one-way street for information. It starts with the boss, the master blueprint. That's your DNA. It holds all the instructions. Then those instructions are used to build the workers of the cell, proteins, that do, well, pretty much everything. And this flow from plan to action, it's happening in every single one of your cells, right now.
Okay, so first things first. Before you can use the instructions, you have to make a copy. Before a cell can divide or do any heavy lifting, it needs a perfect copy of its DNA. And that process is called replication.
I mean, it just makes sense, right? You wouldn't take the original, priceless architectural blueprint to a messy construction site. No way. You'd make a high-quality photocopy. That is exactly what replication is. It keeps the original safe and sound while making a working copy.
And the rules for this biological photocopier, they're actually super simple. It's all about pairing up. A always hooks up with T, and C always links with G. That is literally it. Just 2 simple rules for copying the entire instruction manual for life.
So let's see this in action. Here's a piece of DNA from our worksheet. We read it in these little 3-letter chunks. So the first one is TAC. Okay, let's apply our rules. T pairs with A pairs with T, and C pairs with G. Boom! TAC becomes ATG. Simple as that.
And check it out, we just keep applying that same rule all the way down the line. CGG becomes GCC, ATG becomes TAC. You just follow the pattern. And just like that, we have a complete, perfect copy. Step 1 is done.
All right, we have our copy, but it's stuck inside the cell's main office, the nucleus. We need to get the instructions out to the factory floor, where the proteins are actually built. And for that, we need a new process, transcription.
Transcription is all about creating a temporary, portable message. It's called messenger RNA, or mRNA for short. It's kind of like a quick email sent from the boss with instructions for one specific task. It can leave the nucleus without ever risking the original DNA master plan.
Now listen up, because here comes a little plot twist. For this step, there is one crucial rule change. When you're making RNA, there's no T. Nope, it's gone. Instead, A pairs up with a new letter, U for uracil. The C and G pairing, that stays exactly the same. So the only thing you have to remember here is A now pairs with U. Okay, let's try out this new rule. We go back to our original DNA strand. Remember that first part, TAC? Well, T still pairs with A, but now A pairs with our new friend U, and C still pairs with G. So, TAC on the DNA gets transcribed into AUG on our new messenger RNA.
And if we apply that A to U rule all the way down the line, we get our final message. You can see how the original DNA code has been, well, transcribed. ATG becomes UAC, GAT becomes CUA, and so on. Our coded message is now written and ready for delivery.
And now we've arrived at the final, most amazing step, translation. This is where the magic happens. The coded message finally gets read and turned into an actual physical thing. We're about to go from information to action.
So the cell's machinery reads this mRNA message in those 3-letter groups we've been seeing. They're called codons. You can think of each codon as a word, and each word is an instruction for one specific building block called an amino acid. You just link these amino acids together in the right order, and you build a chain that folds into a protein.
So how do we read the code? We need a special tool. This is it, the genetic code decoder wheel. This thing is basically our secret decoder ring for the language of life. It lets us take any of those mRNA codons and find out exactly which amino acid it stands for.
And using it is way simpler than it looks. Trust me. You just start in the very center with the first letter of your codon. Then you move out to the next ring for the second letter. And finally, you find your third letter in the outer section. That's it. It points you right to the answer.
Let's decode our first one together. Our first mRNA codon was AUG. So we start in the middle at A, move out one ring to U, and then find G on the outer ring. And there it is, methionine, or meth for short. This is a special one because it's often the start signal for building a protein.
Now let's watch the whole changes come together. The next codon, GCC, if you trace it on the wheel, gives you alanine. UAC gives you tyrosine. We just keep on reading each codon and linking the right amino acids together, building our chain one after another, until we get to that very last one. But wait, what happens at UAG?
So that last codon UAG, if you look it up on the wheel, you'll find it doesn't actually code for an amino acid. It's a stop codon. It is literally the period at the end of the genetic sentence. It tells the machinery, hey, job's done, the protein is complete, stop building.
And just like that, you've done it. You have followed the instructions all the way from the master DNA blueprint to a finished chain of amino acids, a brand new protein, ready to get to work. You have officially cracked the code. So let's just boil it down. It's this beautiful three-step flow. Replication makes a DNA copy. Transcription rewrites that DNA into a portable mRNA message. And translation reads the message to build a protein. And when this works perfectly, life works. But a tiny mistake, just one wrong letter, can change the final protein, and that's the root of so many genetic diseases. Understanding this flow is everything.
We walked through that first molecule together, step by step. But now the challenge is all yours. Using the exact same rules we just covered and that decoder wheel, what proteins would these next two DNA sequences create? You've got all the tools now. So, can you break the code?